I've been asked to summarize what is the current status of inflammatory markers in COVID-19 as far as concern of disease severity, prognostication, and outcome is concerned. So, but before we go on to the inflammatory markers, I am a clinical immunologist, so I just take this opportunity to discuss how actually the disease is flourishing and what are the factors, viral or the host, which are actually determining the inflammation and the intensity of inflammation, which is actually causing the major problem in patients with COVID-19 disease. So, uh, actually, we all know that once the virus is entering the human body, or rather the nasal or the respiratory epithelial cells, the virus, once it enters the cell through ACE2 receptors or through neuropilin 1 and 2 receptors, which has been very recently described, it causes activation of toll-like receptors. You can forget it, but I'm just being an immunologist, so I'm going into the details, that which are TLR3 and 7, which are in the phagolysosome, and also through MDA5 and RIG1 complex, which is present in the cytosol. So some of the viruses which are in the phagolysosome will interact with TLRs, and some of the viruses which somehow reaches the cytoplasm will be recognized by MDA5 and RIG1. MDA5 and RIG1 are pattern recognition receptors, and they typically recognizes the RNA of the virus and starts a downstream signaling cascade. You don't need to bother about the downstream signaling cascade, but the net effect of this downstream signaling cascade is that it causes upregulation of interferon type 1 genes. And type 1 interferons are alpha and beta. These type 1 interferons, they in an autocrine as well as paracrine fashion acts on interferon receptors. And through interferon receptors, it causes downstream signaling in the same cell or the paracrine cells through STAT1 and causes upregulation of interferon related or regulated genes. And amongst these, the most powerful gene is your type 3 interferon, which is interferon lambda. And this interferon is actually responsible for killing of the intracellular virus. So this is a normal immune system working in most of us, but some of the patients of COVID, actually they do not mount this type of interferon response, which I will be coming to in the subsequent slides. Virus also knows how to evade the immune system, and for this a corollary has been taken from the original coronavirus, that is a SARS coronavirus, which caused the epidemic in 2002. And some of its proteins, which has been listed out here in this table, are actually known to block various effector pathways of the immune system and thereby antagonizing them, resulting in a very low type 1 interferon response. Now, what happens when they the virus survives? And once the virus survives, it proliferates. When it proliferates, it causes damage to the cell. And when the cells are getting damaged, these cells are actually secreting a lot of damage associated molecular patterns, which are called as DAMPs. And amongst these ATP or heat shock proteins or the nucleic acids in the cell, they are the major sort of attractors of innate immune system, basically neutrophils and monocyte macrophages, and causes their hyperstimulation. Once this hyperstimulation is happening, these neutrophils and the monocyte macrophages, they are secreting a lot of cytokines, as has been listed out here. Predominant is IL-6, IL-2, TNF alpha, interferon gamma also, and macrophage inhibitory factor 1 alpha, and many more. So these cytokines create a cytokine storm. And because of this cytokine storm, the nearby cells, the capillaries, the endothelium, they are getting damaged. and that is why a lung becomes more sort of permeable by damage to the endothelial cells, cause edema, and this inflammatory infiltrate causes damage to the even the airway epithelial cells which are not infected by the virus. So here I had shown that had it been a normal immune response, there would have been a good going type 1 interferon secretion, which could have led to the killing of the virus and minimal damage to the lung. However, and this is probably happening in most of us who are asymptomatic despite having the infection or having a mild COVID disease. However, in patients who have got severe COVID disease, the type 1 interferon response is poor, whereby it is allowing the virus to survive and causing damage to the airway epithelial cells, leading to secretion of DAMPs. And these DAMPs are causing a hyperstimulation of the innate immune system, especially neutrophils and monocyte macrophages. And which, as I have been listing here in this diagram, a lot of cytokines are getting upregulated uncontrollably. How this information has been derived? This has come from some of the very elegant papers which have come out in the very initial part of the when it started in China. And this is study which relates to the transcriptional profile of the COVID-19 patients, elegantly described. As you can see, the interferon beta, which is a type 1 interferon, is low in patients with COVID-19 as compared to the controls. Same way, you can see that some of the pro-inflammatory chemokines and cytokines are high, whereas the ones which protect, like interferon beta, lambda, or IL-1 beta, are low as compared to the controls. So we know that type 1 interferon response is poor. And this has been further consolidated by this study, where they infect the airway epithelial cells with the virus, that is COVID virus, and also influenza A virus. And then they looked at by transcriptomic upregulation of certain genes of inflammatory pathways. And there again, it was clearly shown that there was low interferon regulated genes as well as type 3 interferon expression when the epithelial cells were infected with COVID virus as compared to influenza A virus. Further, it has been demonstrated earlier in the original coronavirus infection that if you infect mice which are knocked out for toll-like receptor 3, 4, and the other downstream signaling pathways, then these mice will develop severe viral replication and cause more pathology in the lungs and consequent morbidity and mortality. Also, it has been, there are many papers. We all know that neutrophils kill the pathogens, any bacteria or virus, by phagocytosing it or by secreting its enzymes which are located in the granules of the neutrophils. But recently, over last say 2 decades, a new method of neutrophil mediated damage to the pathogen has been observed and has been reported in form of netosis. What it does is that once neutrophil is activated by the pathogens and it is unable to control the infection, or it perceives that the danger is too much, it secretes its DNA in form of nets, as a fishing net. We have all seen all these fishers, they try to catch fishes. So these nets are produced out neutrophils into the plasma or the circulation, and it entraps the either bacteria or the viruses. And these nets are actually DNA or histones, and on top of them, they contain the granules, enzymes in the granules of the neutrophils. And whenever these pathogens come in contact with these nets, they are killed. So netosis has been demonstrated to be increased in patients with COVID. Why I'm telling all this? Because we can have a marker in form of cell-free DNA, which tells you the severity of the disease, and there are papers on that. So it is clear that COVID-19 inhibits interferon production at various levels, and there is less of type 1 and type 3 interferon production, which allows the virus to survive and proliferate. So this was about the innate immune system, and neutrophils are actually being super activated because of DAMPs, and they keep on releasing cytokines. Now, generally what happens that after 4 to 5 days of innate immune response, this response is taken over by the adaptive immune system. This is what is happening normally. And we know that T cells and B cells forms the core of the adaptive immune system. Amongst the T cells, CD4 positive T cells and CD8 positive T cells are the most important one. Among the CD4 cells, we know there are certain different subsets which are called as Th1, which secretes a lot of gamma interferon and IL-12. There are Th2 cells which secrete a lot of interleukin 4. There are Th17 cells which secrete a lot of IL-17, IL-22 or IL-23. And there are regulatory T cells which secrete a lot of IL-10 and down regulates the activated immune cells. There are other subsets of T cells which are called as T follicular helper T cells, which provide help to the B cell in causing these B cells to become a plasma cell and to secrete antibodies. I'll come to that later on. So as far as T cells are concerned, both CD4 and CD8 T cells are low. Total T cells are low, and both CD4 and CD8 T cells are low in patients who are severely infected with COVID-19. As has been listed out in this paper, which was published in May, that all the ICU patients have a low total T cells as well as low CD4 and CD8 T cells. Similarly, patients who were of age more than 60 years were having low, and that explains why the morbidity and the mortality is more in this cohort of patients. And also we can see that those who died versus those who survived, the numbers of CD4, CD8 T cells were much low in patients who didn't survive. And these CD4, decline in CD4 and CD8 T cells are actually correlating with the cytokines level. And we have listed out here, in this paper only it has been listed out that there are a lot of cytokines like we can see IL-6. There is a inverse correlation with IL-6, inverse correlation with TNF, and as has been shown here, there is inverse correlation with and the cytokines. So clearly, as the T cells are going down, the cytokines are going up, and these cytokines are playing a major role in the pathology and causing the pathogenesis. Also, in this very paper itself, once immune system gets sort of signals that it has to stop now. So these T cells, once they have become activated and now they have to curtail their activity, will be expressing more of PD1, which is called as program death 1 and TIM3. These are all markers which suggest that these T cells are now probably wanting to stop their effector function. So these we call in immunology as exhausted T cell phenotype. So it has been reported that these T cells are expressing, a lot of T cells are expressing these markers of exhaustion. And there is a very important corollary to these exhaustive phenotype of the T cells, not from the COVID, but from my experience. And there was a paper, elegant paper, which was published 2 years back in 2018 in patients with sarcoidosis and lung fibrosis, where it was reported that CD4 positive and PD1 positive T cells, we generally believe they are exhausted phenotype, but they secrete a lot of TGF beta. And this TGF beta is actually is the major cytokine which causes fibrosis. So as we are seeing sort of a tsunami of patients are coming back to us again after discharge from the COVID hospital with respiratory discomfort or breathlessness and lung fibrosis. So probably these kind of cells might be playing a role. We don't know, but it will be sort of one has to see whether in future we get some of these kind of reports or not. The cytotoxic T cells, which we call as also CD8 positive T cells, they are actually, as we can see here, are highly activated. The activation markers are Ki67 positive or HLA-DR positivity. As compared to mild, the severe patients will have a lot of activated cytotoxic T cells. And once you see that patient with mild disease, they tend to recover over a couple of days. The markers of these CD4 and CD8 T cells, they goes down, whereas those which continue to have severe disease do not show any change in the levels of the perforin granzymes, which are markers of cytotoxic T cells, both CD4 and CD8, and or it may increase. So it can be concluded that total number of CD4 and CD8 T cells are low in severe group, at least around the third week, and a lot of activation markers are expressed on both CD4 and CD8 T cells in severe group as compared to the mild group. In the severe group, over a period of time, these activation markers do not decline. So if we suppose follow patients with these markers, we will know that this patient is likely to go into a severe or a critical illness in times to come, or we need to change our management of immunosuppression appropriately. So this is a summary of T cell mediated immunity in COVID-19. As I had already said, I'll just add one more line that there is a decrease in T regulatory subsets in the severe disease as compared to the mild disease or the healthy controls. Another arm of the adaptive immune system is the B cell. And we all know that B cell will secrete a lot of antibodies, and a lot of hopes and expectations are there with the COVID vaccine, where we are expecting that we will have a good vaccine which will lead to generation of lot of neutralizing antibodies. And as has been listed out in the bottom of this slide, that the antibodies to receptor binding domain, that is S protein, which is also called as a spike protein, is seen in around or more than 90% of the individuals by day 11, whereas IgM antibodies to this receptor binding domain of S protein is seen in to 83% of individuals by day 12, and IgG antibodies to nucleoproteins are seen in almost 2/3 of the patient by the end of 2 weeks. But these antibodies, as you can see, their levels tend to decline with time. So I'll just skip this slide for the brevity. There are basically two types of antibodies. One is a non-neutralizing antibody, and the other one is a neutralizing antibody. So the non-neutralizing antibody binds to certain sites on the virus, and it may lead to formation of immune complex. And this immune complex actually may facilitate phagocytosis by the monocyte macrophages or the dendritic cells. And in contrast to what we believe that immune complexes are cleared, these can cause enhancement of the COVID infection. And this is known as antibody dependent enhancement type of reaction. And this is crucial because there are reports where IgM antibody to the virus are believed to play a role in ADE. Whereas neutralizing antibodies, they either bind to the receptor binding domain at points where the this receptor is actually interacting with the ACE2 receptor or the neuropilin receptor. And there are a lot of portions of these antibodies which has been described in literature. But as we all are learning every day that the antibodies are declining very fast. And there are reports that individuals who have recovered from the coronavirus infection, maybe by day 60 or day 45, there was one report from KEM Hospital in Mumbai, where they reported by day 45 that most of the patients who have recovered from COVID-19, their antibody titers were undetectable. So somehow this viral infection is unique in not allowing our immune system to mount an effective B cell response. And when we say antibody response, we all know as immunologists that it also entails a lot of cooperation from the T cells also. So there is a lot of immune dysregulation whereby both T and B cell are not being allowed to mount an effective immune response. And for this, I'll briefly introduce you to not a very complicated immunology, but B cells, when they are encountering epitope on the antigen of the virus, they go into the nearby germinal center, wherever it is, either in the lymph node or in the spleen. And these B cells, which have are antigen specific, they start expressing this molecule called as BCL6. It is a transcription factor, and BCL6 actually converts these B cells into the plasmablasts, or long-lived plasmablast, or the memory plasmablast. And for this, they need help of the T cells, T helper cells, which are called as T follicular helper cells. And these follicular helper T cells, they also express BCL6, as I have been showing here in this particular diagram. This is taken from another paper from Proceedings of National Academy of Sciences in North America, where it shows that the T cell, once it becomes activated by the dendritic cell or the antigen presenting cell, it goes into the germinal center and starts expressing a lot of BCL6. So these BCL6 positive T cells, when it gives help to the BCL6 positive B cell, these B cells become long-lived B cells. They convert into plasmablast, long-lived plasmablast, which secrete high-quality, high-affinity antibodies. Now, if suppose this reaction is not happening, and this B cell or this T cell is not expressing BCL6, and it is activates the B cell here, as you can show here, it will lead to short-lived plasma cells, and therefore the antibody response will be of a shorter duration. So probably this is somehow defective. The expression of BCL6 is being defective in the patients with the COVID-19, especially those who are having either severe disease or are having antibody responses which are of short duration. And for this, I'll quote a very elegant paper published in Cell in the month of August. Here, the authors have taken out the during the postmortem only. They have taken out thoracic lymph nodes and the spleen of the individuals who died of severe COVID disease, and they looked at the expression of BCL6 by immunohistocytochemistry. And you can see that in the healthy non-COVID-19 lymph nodes, you can see a lot of BCL6 is getting expressed, which is overlapping with CD3 positive. CD19 B cells are CD19, CD3 is for T cell, CD19 positive B cells. This is the overlap image. However, in patients who were COVID and who had died by the end of 3 weeks, you can see almost negligible expression of BCL6 in the thoracic lymph node, and there is no overlap with the B cells. They looked at patients who died early within 7 days after admission, and again they found the same, that the BCL6 expression was much less as compared to the non-COVID. You can see the BCL6 expression here, and you can see the BCL6 expression here. Hardly any. As has been representative, a bar diagram has shown that COVID-19 patients who died late or early, they had a very limited or very little expression of BCL6 in their lymph nodes. Then they go on to look at the B cell expression or this BCL6 expression in their spleen also. And again, they couldn't find much of expression as compared to the control. Hardly there is any expression of BCL6. SP means spleen, the tissue is splenic. And then they go on to look at the expression in the late patients who died late also. They couldn't find any expression of BCL6 in the spleen of these individuals, in the B cells. So clearly the B cells are lacking BCL6. And similarly, they had shown that T cells were also not expressing the BCL6. So here they have stained with CD4 and BCL6, and when you see overlay, hardly any expression of BCL6 as compared to the controls where you see a lot of BCL6 expression, and a lot of it is getting sort of expressed by the CD4 positive T cells also. So clearly showing that CD4 positive T cells are also not expressing BCL6 in patients of COVID-19 who have died either late by 3 weeks or early, that is by 7 days. And same they could demonstrate in the screen also. So the reason why I'm actually delineating all this out is that obviously we are not going to do BCL6 in the expression in the T cells or the B cells, but we should understand why the antibody response in patient with COVID-19 is actually not there as it should have been or as it has been in many other viral infections. Now this portrays a sort of a very grim picture. Why? Because if the T cell and the B cell responses are poor, then chances of getting protective immunity after one infection are that this immunity may not last for a long period of time. And then you may get either a reinfection from the same strain or from a mutated strain of the virus. So the conclusion is that defective BCL6 explains the mechanistic part of the limited durability of the antibody responses in patients with severe COVID-19. We all are hearing every day, and there are actually in May there was a lot of enthusiasm when the postmortem studies of some of these patients were published that it is not an inflammatory disease, rather it is more of a thrombotic disease. But actually both thrombosis and inflammation are interlinked, because we all know the virus has a tendency to go through the ACE2 receptors into the capillary endothelium cells also. And once it goes there, it damages the endothelial cells. And once the endothelial cells are damaged, the basement membrane collagen or tensin C are exposed. So once this basement membrane collagen and tensin C are exposed, platelets become activated. And once the platelet become activated, they start forming a platelet plug, secreting a lot of fibrinogen and von Willebrand factor, which promotes thrombosis. So, and also platelets are activated by secretion of lot of pro-inflammatory cytokines by the neutrophils and the monocyte macrophages, including interleukin 6, interleukin 1, TNF alpha. They all cause activation of platelets, so which promotes thrombosis. So these inflammatory or immuno-inflammatory thrombotic events actually are the hallmark of the pathogenesis of COVID-19. So I just try to list here some of these markers for the sake of absolute clarity that there is endothelial injury by the virus and also there is high plasminogen activator inhibitor 1. So the clot lysis mechanism is disturbed and patient is actually in a state of hypercoagulability, which promotes thrombosis. Similarly, platelets are active, so they release a lot of pro-inflammatory and the thrombogenic mediators, which promotes thrombosis and cause microthrombi. So this is the total part of the immune system that I wish to discuss today with you, that this is a beta coronavirus, we all know, and virus enters through ACE2 receptors and also neuropilin-2 receptors, which have been recently described. Normal immune response in form of type 1 interferon, if it is there, or T cells, B cell responses are normal, it will clear the virus. However, in few unlucky percentage of patients, virus evades the immunity by inhibiting the interferon axis. We don't know why exactly, which leads to a persistently activated state of the innate immune system, which causes a lot of pro-inflammatory cytokines being secreted in form of IL-6, IL-1 beta, TNF, and also activation of inflammasome and netosis by the neutrophils. The adaptive immune cells are downregulated, both T and B cells, whereby T cell responses are also not there and the B cell responses are poor, and they are unable to mount an effective immune response. And this ultimately results in hyperinflammation or cytokine storm, which causes more problem by damaging the capillaries and causing the organ damage. So if there are any questions till now, because now after this I will go on to the lab parameters which we can use day in, day out for monitoring the patients, and which we should be using and why with the evidences. So if there are any questions, or if you allow me, then I go on and discuss the next part of my presentation, that is the lab parameters to watch for during management of COVID-19 patients. And I will be talking about the evidence-based sort of parameters which are there. So this is a study published in Nature Communications, which is a cross-sectional study of immunological and inflammatory markers. This study was published as early as April 2020, and this is from China, one of the hospitals in Beijing. And here, as has been listed out here, this is a study which is being studied on 41 COVID-19 patients, 29 with mild disease and 12 with severe disease. There was two patients who had actually died in this cohort also. So that is there. And as it can be seen here, the patients who had got severe disease, their T cell, CD3 positive T cell responses are poor. Same is true for their CD4 cells and CD8 cells. The B cell count was normal. However, the NK cell, which is a natural killer cell, the population was much less. Rest of the frequency of CD3 positive T cells, they were all normal. So, except that the frequency of B cells was increased in compared in the severe cases as compared to the mild. Then they looked at the activation of the T cells. The activated T cells were similar in both mild and the severe group, though there was some suggestion that the severe group had a higher levels of activated T cells, whether they are cytotoxic T cells, CD8 positive T cells, or CD4 positive T cells. Also, they looked at the exhaustive phenotype, that is PD1 positive CD8 cells and PD1 positive CD4 cells. They were almost similar. However, CD8 positive, PD1 positive T cells were a bit higher between mild versus severe, whereas CD4 positive T cells, which were PD1 positive, were almost similar in both the categories. So there is aberrant activation and dysregulation of CD8 positive T cells in patients with severe COVID-19. Probably it might be playing an important role in the pathogenesis of the viral pneumonia or ARDS. From the same study, which shows clearly that patients, this is basically as per the severity of the patients. The higher the number, that more severe the disease. So the levels of interferon alpha were much less, the mean levels were much less as the disease severity is increasing, as can be seen by following these lines here. Same is true for interferon gamma, which is actually rising in patients who have got severe disease, and CXCL10, which also rises when the disease is severe. Whereas the coagulation markers, they have looked at 3 coagulation markers: D-dimer, angiopoietin 2, and von Willebrand factor A2. It can be seen that the levels of D-dimer increases with the severity. Same is true for the levels of angiopoietin 2 and also the von Willebrand factor. And when they looked at the levels of some of the inflammatory markers like IL-6, GM-CSF, and uh, basically your RAGE proteins, it can be seen that levels keep on increasing with the severity of the disease, especially of IL-6 and GM-CSF. So coagulation and inflammatory markers are going almost in concordance with each other. Another study which came out very early, and this was a meta-analysis of 7 studies or 8 studies which were actually available at that time. And it can be seen here that the level of CRP was increased in patients with severe group. And this is the forest plot, and this is the total analysis being given here. That there was a difference between severe versus non-severe as far as CRP is concerned. Same is true for your ESR. It is increased in severe group in favor of severe group. Same is true for serum amyloid A protein, which is also an acute phase response protein. Same is true for ferritin, IL-6. Though the level of procalcitonin were shown to be increased in patients with severe group, but the subsequent studies have not confirmed whether procalcitonin can be used as a prognostic marker or is to identify differences between severe and non-severe group. In another study where they have used the levels of, and this is again a meta-analysis, and this is published I think in the month of May, and here they looked at patients of COVID-19 and try to identify what are the markers which actually suggest that patient is going to have a severe disease. So again, the number of WBCs or total leukocyte count, as simple an investigation as we can think of, which is available everywhere in the world, shows that the higher number of CDC, total leukocyte count actually favors severe disease. And patients who presented with an increased number of WBC of more than 10,000 had a 3-fold higher risk of developing severe disease, and the combined odds ratio was around 2.92 in this particular meta-analysis. Similarly, we can follow neutrophil lymphocyte ratio, which is again a very simple investigation available throughout everywhere. You don't need any expert laboratories. Clearly, it establishes the fact that the elevated levels of neutrophil lymphocyte ratio are associated with disease severity. And it could be a predictor of disease progression. In this particular meta-analysis, also authors have reported that procalcitonin actually identifies is high in patients with severe disease. And same is true for the CRP. So clearly establishing the fact that immune inflammatory parameters such as total number of white blood cells or total leukocyte count, lymphopenia, neutrophil lymphocyte ratio, procalcitonin, and CRP correlate with disease severity and can be used as a marker to assess or to follow patients much more closely who are likely to develop severe disease. Okay. Then this was another sort of a meta-analysis which is published, I think, in the month of August, where it was shown that in Asian population, and again, this is the study from China predominantly. So again, here also in this meta-analysis, the number of neutrophils and the increase or the higher levels of erythrocyte sedimentation rate clearly favors patient in the severe group. Same way CRP and also lactate dehydrogenase. So I like to have a few word about lactate dehydrogenase here. Lactate dehydrogenase is either a marker of uh, sort of ongoing thrombosis or damage to the cells or the organs. So clearly, if the lactate dehydrogenase levels are high, then the patients are likely to have severe disease. And also, the levels of IL-6, though they were not found to be significantly differentiating between patients of severe versus mild disease. And in this very study, procalcitonin was not found to be, rather, it was found to be much more higher in patients who have got severe disease. Also, the levels of fibrinogen, which were high in patients with severe disease, as well as levels of D-dimer, neutrophil lymphocyte ratio, and lymphocyte CRP ratio, which is again a very simple prognostic marker and is available to most of us in our hospitals, can actually differentiate patients between mild versus severe. So this is a pool table of the same study where they have listed out that total number of leukocyte count, neutrophil count, lymphopenia, low platelet count, increase in prothrombin time, elevated levels of D-dimer, elevated levels of fibrinogen, ESR, procalcitonin, and CRP, and also pro-inflammatory cytokines like IL-6 and anti-inflammatory cytokine like IL-10, and the ratios of CD4 and CD8 T cells can differentiate between mild versus severe disease. Not only between the severity, but also if you have a high WBC or high neutrophil count and lymphopenia, which is persistent, then these patients are likely to have an adverse outcome in form of non-survival or mortality. And same is true for platelet count. Generally, we have seen that patients, when they are active, they have a high platelet count. But in patients who have got a poor outcome, they have a low platelet count. And same is true for prothrombin time. Elevated prothrombin time or bleeding diathesis can actually be seen in patients who are likely to die. And same is true for the other activation markers like D-dimer, ferritin, ESR, procalcitonin, CRP, and IL-6, which actually are sort of forerunner of an adverse outcome, especially mortality, if their levels are too high or persistently high despite treatment, and if they remain elevated for 7 or more days. So several parameters like WBCs, lymphocyte, platelets, which are available everywhere, prothrombin time, D-dimer, and fibrinogen, even if there are no availability of IL-6 or IL-10, but can make you guide the or tell you about the severity of the COVID-19 disease and the likely outcome of this particular patient. So if patients, God forbid, have these kind of adverse investigations, then these patients need to be followed much more closely, and we need to design some more immunosuppressive or alternative form of treatment so that we can make these patients survive. And this is another study from again China, and this will just be the repeat of the same data which I had repeated earlier. The difference between the survivor versus the non-survivor. And as has been shown that the number of neutrophil count, the platelet count, if they are low, are actually telling you that these patients are likely to die. And same is true for the coagulation parameter marker. If you have a derangement in the coagulation parameter marker, then the chances are that these patients will die. And same is true for the inflammatory markers, that if they are persistently high, then these patients are likely to die. As has been shown here, the CRP and the procalcitonin, IL-1 beta, IL-8, and IL-2 receptors. So this is a table summary of potential laboratory markers and their significance during this pandemic. If you have lymphopenia, obviously we have seen that T cells, B cells are all low. If you have leukocytosis, especially in form of neutrophilic leukocytosis, this could be part and parcel of severe COVID disease, or there could be on top of it a super infection with a bacteria. Now that becomes a real challenge because not very frequent culture facilities are available in most of the COVID hospitals. One should make any attempt to diagnose these patients, or even if required, empirically treat these patients with broad spectrum antibiotics. Patients who have got thrombocytopenia or even say neutropenia or total leukopenia, these patients should be thought of heading towards a possibility of macrophage activation syndrome, and these needs to be followed much more closely, especially with the level of the serum ferritin. So given the whole of the spectrum of all the markers and their clinical relevance in form of this table, you may go through it. I know all of you know about it very well. And this is almost the penultimate slide of my presentation, where this paper, which is published in Nature Medicine in the month of August, for the very first time, very clearly gives you the total levels of cytokines. If somebody is interested in assessing the outcome as far as level of cytokines are concerned, so this is the study that needs to be followed, where they have given a level of more than 70 for IL-6, more than 50 picogram per ml for IL-8, and more than 35 for TNF-alpha, and more than 0.5 for IL-1 beta. And it has clearly shown that patients who have got severe disease will be actually having a poor outcome. As has been shown here, the survival, the graph shown on the bottom of the slide, that the survival probability decreases as the levels of IL-6, IL-8, TNF-alpha, and IL-1 beta are high. And these cytokine levels correlate with the disease severity and independently predict survival, as has been shown here in the Kaplan-Meier curves. We can see that the high levels of cytokines are actually associated with poor outcome, both survival as well as oxygen saturation, and obviously they denote the severe disease. So with this, I'd like to end my presentation, and before that, I'll definitely like to thank my colleagues, especially Durga, who is the assistant professor in my department and is the real force behind. He had made me understand some of these very facts. And Mohit, who is my PhD, as a young scientist in my lab, who has made some of these very basic concepts clear to me because I was not able to understand some of them. And also my senior resident, third year, Naveen, who had helped me in actually designing some of these studies. And also my second year, Anamika, PhD scholar, who has actually synthesized few slides for me and made them understood to me. So the take-home message is basically there are several laboratory parameters as simple as total lymphocyte count, platelet count, prothrombin time, D dimer, and fibrinogen levels. If availability of IL-6 and IL-10 is there, maybe. Otherwise, you do not need them. And CRP could be telling you about the severity and the actual outcome in the patients. So if available, kindly screen your patient with these investigations and keep monitoring them for the progression of the disease and take steps accordingly. Thank you. And if there are questions, I will try to answer.